welcome back to this chapter on forensic science. Now we are covering chapter 8, the microscope, as we look into the textbook Criminalistics, an introduction to forensic science. Introduction. A microscope is an optical instrument that uses a lens or a combination of lenses to magnify and resolve the fine details of an object. The earliest methods for examining physical evidence relied solely on the microscope. The virtual image is the magnified image seen by looking through a lens. A real image is an image viewed directly by the eyes. The object to be magnified is placed under the lower objective lens and viewed through the upper lens known as the eyepiece. Various types of microscopes are used to analyze forensic specimens. The principle of a compound microscope, the passage of light through two lenses, forms the virtual image of the object seen by the eye, reflecting through multiple lenses, eventually to show the image that is being observed, but magnified. The compound microscope. In the basic compound microscope, the objective to be magnified is placed under the lower lens, and the magnified image is viewed through the upper lens. The magnification of the image can be calculated by multiplying the magnifying power of the objective lens by the magnifying power of the eyepiece. The microscope is composed of a mechanical system which supports the microscope and an optical system which illustrates the object under investigation and passes light through a series of lenses to form an image. With a lot of microscopes, usually the eyepiece that we look into has a ×10 magnification, and then toward the bottom of the microscope there is some adjustable lenses, usually often 4x, 10x, even up to 40 and 100. So if you have an eyepiece of 10 and you are looking through an objective lens of 4, 10 times 4 is 40 times the actual size of the image. Oftentimes we don't need to get to 1,000 unless we're looking at organelles of microscopic organisms. So here are just the various pieces of a compound microscope and how light travels through them. The mechanical system includes the base, which is the support, and the arm, which is the C-shaped upright structure. And whenever a microscope is carried, it's always proper to have one arm on the base, this long vertical portion, and one hand under the base, and one hand along the arm to best protect the inner workings. Then there is the stage. That is the plate on which the specimens are placed, right around here. The body tube is the hollow tube on which the objectives and eyepiece lenses are mounted. There's also coarse adjustments and fine adjustments. Those are separate knobs. The coarse adjustment knob is used to focus the microscope lens by moving the body tube, while a fine adjustment is used to focus the lens by moving the body tube, but to a very smaller magnitude. Usually you start with coarse adjustments first, and then when you're around where you need to see your organism, then you use fine adjustments. There's also the optical system, known as the illuminator, the artificial light source used to usually supplied by a light bulb to illuminate a specimen from underneath. Transmitted illumination, when the light is directed through the specimen from the base, and vertical or reflected illumination, when the light comes from above and reflects off of the specimen. There is the condenser, lens systems under the microscope stage that focuses light onto the specimen. Objective lenses are the lenses closest to the specimen. Usually several objectives are mounted on a revolving nose piece. And then there's the parafocal, when the microscope is focused with one objective in place, another objective can be rotated into place and the specimen remains very nearly in correct focus. Eyepiece or ocular lenses. The lens is closest to the eye. The monocular, it can be monocular, a microscope having a single eyepiece, or binocular, a microscope having two eyepieces. In the case of binocular microscopes, it's best to move the eyepieces so that both eyes can look through the sample at the same time. The comparison microscope consists of two independent objective lenses joined together by an objective optical bridge to a common eyepiece lens. When a viewer looks through the eyepiece lens of the comparison microscope, the objects under investigation are observed side by side in a circular field that is equally divided into two parts. Firearms. Modern firearms examinations began with the introduction of the comparison microscope, with its ability to give the firearms examiner a side-by-side magnified view of bullets. Here we're looking at a comparison microscope, two independent objective lenses joined together by an optical bridge. The stereoscopic microscope is actually two monocular compound microscopes properly spaced and aligned to present a three-dimensional image of a specimen to the viewer who looks through both eyepieces. Particularly useful for evidence not requiring very high magnification, usually times 10 or 125 times the original size. Large working distances make it quite applicable for examining big, bulky items. And here's a schematic of a stereoscopic microscope. This microscope is actually, again, two separate monocular microscopes, each with their own set of lenses, except for the lowest objective lens, which is common to both microscopes. Then there is polarizing microscopy. Light that is confined to a single plane of vibration is said to be plane polarized. The examination of the interaction of plane polarized light with matter is made possible with the polarization microscope. Polarizing microscopy has found wide application for the study of bifringent materials. Materials that split a beam of light in two, each with its own refractive index value. Determining these refractive index data provides information that helps to identify minerals present in a soil sample and the identity of a man-made fiber. The microspectrophotometer is a spectrophotometer coupled with a light microscope. The examiner studying a specimen under a microscope can simultaneously obtain the visible absorption spectrum or infrared IR spectrum of the material being observed. The instrument is especially useful in examining a trace evidence, paint, fiber, and ink evidence to help identify what those consist of. The scanning electron microscope it bombards a specimen with a beam of electrons instead of light to produce a highly magnified image from 100 to, I believe that's supposed to be 100,000 times magnification. Its depth of focus is some 300 times better than optical systems at similar magnification. Bombarding the specimen's surface with electrons normally produces X-rays emissions that can be used to characterize elements present in the material under investigation, determining presence of certain metals or other non-metal elements within a sample to help identify what's being looked at. Here we have a schematic diagram of a scanning electron microscope displaying the image of a gunshot residue particle. Simultaneously, an X-ray analyzer detects and displays X-ray emissions from the elements of lead, antimony, and barium present in a particle.
So as electrons are beamed onto a surface, this excites, and then once this excites the material, the atoms in the material, as light is released, as those atoms relax from excitement, that light is released in the X-ray spectrum, X-ray range, which then can be analyzed by an X-ray analyzer to look for the identities of the different compositions. Primer residue on hands. The firing of a weapon propels residues toward the target, but also blows gunpowder and primer residues back toward the shooter. Traces of these residues are often deposited on the shooter's firing hand. Detecting these residues can provide valuable information as to whether or not an individual has recently fired a weapon. With the aid of a scanning electron microscope, examiners measure the amount of barium antimony on the relevant portion of the suspect's hands, such as the thumb, web, and the back of the hand and the palm. Examiners may use a scanning electron microscope to characterize the morphology of particles containing these elements to determine whether or not a person has fired, handled a weapon, or was near a discharged firearm. Forensic palynology involves the collection and examination of pollen and spores collected with crime scenes, illegal activities, or terrorism. The microscope is the principal tool used in this field of forensic palynology. The information gained from the analysis of pollen and spore evidence has many possible uses. Link a suspect or object to the crime scene or the victim. Prove or disprove a suspect's alibi. Include or exclude suspects. Track the previous whereabouts of some item or suspect. And indicate the geographical origin of some items. Well, just past the 12-minute mark, that concludes Chapter 8 of our forensic science study. Look forward to seeing you all in the next video. Peace.